And today we will continue our conversation about the pipeline, right, in Seurat. The reason we chose it to teach this class this year and as a redesign of the course is because I think it connects many statistical concepts and techniques into one context, into one analysis. So you can just work on one data set and implement those techniques instead of switching tasks. So today we are going to talk about linear dimension reduction, or more specifically, principal component analysis. So before we start, may I ask how many of you have ever used PCA before? Okay, so everyone. Okay, then maybe I can ask one more question. How many of you know how PCA works? Like, what's the purpose of using PCA? What do you expect to get out of it? Okay, fewer hands. Okay, how many of you know the linear algebra behind PCA? No. Okay, so then we'll have something to do today, right? So I think knowing how to use PCA doesn't mean that if PCA does not work for you, you know how to modify it or change it to something else that work better for your data. Okay, so all right, so what we have done last time was last lecture was to about the identifying of highly variable genes. As a very quick recap, why we want to do this. The reason is that we want to choose fewer genes than we have. For example, we know that in our human genome, we have a total of about 20,000 genes. But in the last step of choosing highly variable genes, also called feature selection, we only want to keep the around 2,000 genes. That's a default number in Seurat, but I don't know whether this number has any theoretical justification yet. Probably empirically, people try different numbers, but this is another question. How many should you keep, right? 2,000 compared to 20,000 is 10%. And you choose the genes that have the largest variances compared to their means. So if you recall, we fitted in the default VEST method in Seurat, we fitted a log variance versus log mean curve, right? And the variance and mean are about the count data, not the normalized data. That's the default. And that was another question we had last time. Then we use that curve to decide which genes log variances are much bigger actually than the log compared to the expected log variance given by the mean. So once we have chosen those genes, then we go back to the normalized data in the previous step and use those data for each gene to do z-score calculation, basically. We standardize each gene's normalized values to have mean 0, standard deviation 1. So these 2,000 genes that you keep will have the same scale, same standard deviation as 1 before we go to linear dimension reduction. Okay, so that's where we are today. So PCA stands for Principal Component Analysis. So please be careful about this spelling of principal, because some people, I've seen cases that had this term, it's an adjective misspelled as PLE. That's a different word. But yeah, just be careful. Here, the principal component means it's something major, like a major kind of component. And specifically, it's called linear dimension reduction because each principal component is your new dimension, is your new feature. It's a linear combination of your original features. So this is a very intuitive cartoon from Wikipedia. So here you have two original features. So if you use our context, our language, every feature is a gene. We have two genes, and every dot here is one cell. We're plotting it. So clearly, you will see these two genes have a positive correlation, right? When one gene's value is higher, the other gene's value tend to be higher. So what PCA does, you can consider to be a rotation in this case. Because so let's forget about the scaling part for now. Let's just look at the rotation because that's more intuitive. So what you want to have is that we want the first PC to explain the most variance in our data. So if we look at all possible directions in the 2D space, which direction can give us the largest variance in the data is this direction, right? So this axis, this long axis, because you can see that in this axis, this direction, the data are more scattered more broadly. And that's why we call this bigger variance. Compared to the original feature 1 or feature 2, this PC direction has better variance, has larger variance in explaining your data. So this is where linear algebra gives us. It will tell us that this PC1 is a linear combination of feature 1 and feature 2 with these weights, 0.866 and 0.5. So you can see that feature 1 contributes more to PC1 than feature 2 does, right? This is also something we can see visually. You will basically see that if you look at the angle between PC1 and feature 1, the angle is smaller than the angle between PC1 and feature 2, which means that PC1 itself has stronger correlation with feature 1. And this is reflected in the larger weight. Okay, and something we will also require is that the two weights should be a unit vector in terms of rotation. By a unit vector, we mean that the square of this weight and the square of this weight add up to 1. That's something we will require. Otherwise, you may be, for example, you can multiply these two values by a factor, say by 2 or by 3, then you get the same direction, but you would change the linear combination. So that's what we call unidentifiable. So you could have multiple values give you the same direction. That's why we restrict our rotation to be a unit vector whose sum of squares is equal to 1. Okay, then for PC2, right here, it's orthogonal to PC1. So this is another thing you should learn from. You can learn from linear algebra is that by orthogonal, it means that if we do the dot product or inner product of two vectors, it's 0. You get a 0 product. So this is why you see this value multiplied by this value plus this value multiplied by this value. Keeping the signs, you will get 0. You get cancellation. And so PC2, similarly, is explained more by feature 2 than feature 1. But if you keep the sign into consideration, feature 2 is positively correlated with PC2 is positively correlated with feature 2, but negatively correlated with feature 1, right? This is reflected in the sign. So this positive negative correlation, or the positive and negative weights here, also have a meaning. So when feature 1 has a positive weight in PC1, it means that they are positively correlated. And when feature 1 value increases, PC1 increases. So it's a way for us to interpret the meaning of each PC, because PC is our new feature. So once we do PCA, we will use PC1, PC2 instead of the original feature 1 and feature 2. Another important result to know is that if you keep your PC number as the same as your feature number, you don't lose any information because it's just a rotation of your data. Everything is still kept in your PC new space. Okay, so we will care about the values of these weights and also the signs of these weights. Very soon, we will introduce a jargon for the name of these weights called loading. So this is the loading for feature 1 in PC1, loading for feature 2 in PC2, and the loading can be positive or negative. So basically, what I said is that once you get the two PCs, essentially, you are working in this new coordinate system by rotation. So PC1 is your new dimension 1, PC2 is your new dimension 2. The good interpretation is that your data points have the largest variance in the first PC, and then second PC will explain most of the remaining variance, and the two PCs are uncorrelated, they are orthogonal. So therefore, the variance is explained can add up. So that's the thing. All right, so now why do we want to perform PCA for single-cell RNA-seq? That's another question. I think this is something we should all ask ourselves before we use a method. Why do we want to do this? Why is it necessary for our scientific goal, right? Previously, we said, okay, if we do a very quick recap, we filter out cells that are likely droplets, that are likely empty droplets or doublets. We filtered out the droplets with too high mitochondrial percentage. That filtering step is just to improve our data quality because we don't want to focus on the bad quality cells, right? Then we do a normalization. Why do we want to do normalization? Because we know that a gene's counts in different cells, if we directly compare the counts, it might not be biologically meaningful because it's depending on or it's influenced by the total number of reads or UMIs in the cell. But we may not care about that. If we care about the relative expression of all genes in one cell, we should normalize the cell library size to be a constant, right? That's why we do 10K, but we have the 10K choice and the million choice, and then the 10K choice. And then after that, we do log 1P is actually justified by the variance stabilization perspective. So you want to make the variance not depending on the mean so that we can apply techniques like Gaussian distribution. So this is what we'll explain today. So we hope that we will use normal Gaussian distribution in the next step. So that's normalization. But as we said, there's no gold standard for normalization. You just have to justify it from a perspective, from a criterion. Then after that, we want to reduce our dimensions and also remove what we think as the noise. Then we do highly variable gene selection, right? And then we do scaling for the purpose of PCA. So every step should have a justification. If not, we should ask, why do we want to do this? So for PCA, why do we want to do it? Again, I think we want to work in a smaller dimension. And we know that many genes are highly correlated. So let's look at this. One way to think about it is that seeing that the two original features are highly correlated, if you believe the signal is just their common thing, then you might think, okay, I only need to keep PC1 and I reduce my number of dimensions from 2 to 1. So then I just work with my data projected to PC1. So basically, you just look at the data in the first PC in the horizontal way here, and you consider PC2 to be noise. If that's what you believe, then using PC1 only is considered as denoising. You are throwing away noise but keeping the signal. And also you reduce the dimension. So coming to single cell data, I think this is why we do PCA, because even after highly variable gene selection, we still have 2,000 genes. But do we really believe that we need a 2,000 dimensional space to explain our cell-cell relationship? Essentially, I mean, let me ask this question. What is the most important analysis, or what do people want to get out of single cell data when they measure thousands, tens of thousands of cells together in one experiment? What do they want? Because we have a very early cartoon diagram showing that you want to do cell type annotation, right? In my opinion, I think what people really want to get out of it is to know what are the cell types there and whether the cells have certain interesting relationship. It's like the discovery of cell-cell heterogeneity, because we don't know that before. When we measure all cells jointly by smashing them and combining their RNA with bulk RNA-seq, we lost that heterogeneity part. But with single cell, now we are able to capture that, right? So therefore, if we are interested in studying cell-cell relationships, we may not need such a high dimensional 2,000 dimensional space. Because one thing I hope to share with you is something which I mentioned already, I think, curse of dimensionality. So basically, when your dimension is very high, Euclidean space will be problematic. So just an intuition is that think about a two-dimensional, circle versus a three-dimensional ball. If we set the diameter to be equal to 1 in both the 2D and 3D, and if I draw 1,000 points uniformly from the circle or the ball, how would the points be distributed like within the circle 2D and the ball 3D? And I can tell you that mathematically, The higher the dimension you have, you can go from 3D to 2000D, you will more likely have the points far away from the origin because you have more area, you have more volume in the ball. Actually, I shouldn't say that because a uniball, I shouldn't say more area. That's right. But basically, you will have this tendency that points will become farther away as your dimension goes up. So that's one way to think, to try to intuitively see the cursive dimensionality. So the short message is that if you can keep the majority of your variance, which you consider to be signals, the variance is contributed by signals in the lower dimension, you are in a better position. And that's what PCA aim to do. So we hope that after the PCA, we can reduce the number of dimensions from 2,000 highly variable genes to maybe 20 to 50 PCs. That's a very big reduction of dimensions. And we hope that we still keep most of the variance and which we consider to be signal. So I think you can see the connection between the things we talk about. And the Stevens paper, Nature Genetics paper, talk about this expression model, measurement model, together give you observation model, right? So in that decomposition, expression model is the signal variance, while measurement model is the noise variance. And when you write it out explicitly, you have a decomposition already. Here we don't have a model when we do PCA, but we're just believing that the top PC explains the signal variance, while the bottom PCs explain noise variance. But that's an assumption. So that's why I hope you are aware of what we are making, what assumptions we're making here. Okay. All right. Then this is from Surratt. The step, perform linear dimension reduction. So only the previous determined variable features, that is, the highly variable features, are used as input, but can be defined using features argument. So in the ROM PCA function, there's a features argument if you wish to choose a different subset. However, it says, if you do want to use a custom subset of features, make sure you pass these to scale data first, because the highly variable genes from find variable features, the previous function, was already scaled, right? The other features, if you want to select here, but they were not in the variable features, they are not scaled. So this is just a message for your attention. And then for the first principal components, I think it should be first few principal components. Surratt outputs a list of genes with the most positive and negative loadings, representing modules of genes that exhibit either correlation or anti-correlation across single cells in the data set. So basically, in this command, we are just using the highly variable features identified after scaling to do PCA. And once we get a result, so once you do this, Surratt will store the PCA results into a field called PCA. So you will have this new field in PBMC object. And once you run this print, it will print out the PCA results like this. But you have two things to specify. One is that how many PCs you want to output. Here it's 5. And then how many top features, top in terms of the loading, absolute values, do you want to output for the positive loadings and negative loadings for each PC? So here it's set to be 5. Then for each PC, the top 5 positive loadings, the genes have the top 5 positive loadings are reported. Also, the genes have the top 5 negative loadings. So it's just for your interpretation. Basically, how we look at it is that, okay, I know if PC1 is the most important one that explains the majority of variance in my data, I see it's correlated with this gene, positively correlated with this gene, which means that it has a positive influence on this PC, and negative correlation with this gene, which means it has negative influence on the PC. But one thing we should notice that in some analysis, especially if your data is not normalized, if you're using just log1p count, you're using original count, not the normalized count, you would observe that this PC1 is strongly correlated with cell library size. So in other words, if we go back to here, if we still consider every dot to be one cell, Then the cells that lie on the far right end, which means the cells have the largest values in PC1, are the ones with the largest cell library sizes. And people think, okay, this is not something we want. This is another way to check whether you have effectively removed your library size effect. Okay, so Seurat has offered some very nice visualization functions for PCA. So this is called vis dim loadings, right? Visualization dimension loadings. And here we're just plotting, when you said dims 1, 2, first two PCs. And here, because Seurat can have other dimension reduction methods, so the default is PCA. We use PCA as the reduction method. So essentially, the PC1 plot, loading plot, should agree with this. So these are the features genes on the positive side. They have positive loadings. You see, this one, CST3, has the largest positive loading. TYROBP, second largest. LST1, AIF1, FTL. Top 5, let's go back. These are exactly the 5 genes here, the 5 genes with the largest loadings. But here, negative loading, it only shows 1 gene, MALAT1, which is here. So the plot is not showing everything because we have 2,000 highly variable genes. You could actually have 2,000 rows here, but that will be too long. So that's why it's just showing the top ones, I think, in terms of absolute values of their loadings. So you only have 1 negative appear. And this is PC2. You will see these are the positive genes, negative genes, and they should agree with what you have here. So I think this is just a visual thing to let you know, okay, how my PC1 is determined by my original genes. And sorry, I couldn't understand you very well. Can you speak louder? Yeah. But we're not in a regression setting. We don't have a response here, the Y variable here. It's just unsupervised. Everything we have so far is just this X matrix, right? We have in Seurat format, we have the rows as genes, columns as cells. So we don't have a Y here. Yeah. Okay, so any more questions here? All right. And Seurat also have the dim plot, which is another way to look at the PCA result. But here, every dot is 1 cell. So you're just showing your cells in the 2 PCs, top 2 PCs, PC1 and PC2. And as you can see, we see 3 major clusters, I would say, probably 3 major clusters in our cells. But later, as you will see, people use nonlinear dimension reduction methods like t-SNE and UMAP to see the cells because PCA will not reveal some subtle cell types if they are different cell types, but in PCA, they may be together. They may be seen in one class, one big cluster. And the reason, short answer, is that we have PCA designed to preserve global variances. So it's not really emphasizing local structure. If you want to emphasize the local clusters, you need to go nonlinear dimension reduction. Basically, it's a distortion of your data. And this is like kind of a faithful representation of your data. And also, you can imagine that given only 2 PCs, it's reasonable that we are not seeing some separation, right? Because we originally have 2,000 dimensions, but we go down to 2. It must be that much information is lost. So, but this is the traditional PC plot. But in some other fields, I can think of genetics, PCA, PC1 versus PC2 have been very popular for showing the data. Especially, I don't know if you have seen a very interesting figure, and it was a 2001 Nature paper, probably by John November. He used to be a faculty on UCLA, and then now a faculty in human genetics at U Chicago. So his first author paper have this very interesting PC plot. Every dot is one individual. He used the SNP genotype data as the input into PCA. So it's like individuals by SNPs. Every dot is one individual, and the SNPs are collected by SNP array from people's blood sample, right? Measure the DNA SNPs variants. And then he found that doing PCA on this matrix and give the top two PCs, the positions of individuals in the PC plot can agree very well with the geographic regions they're from. So you can see it's like a map of Europe. Some people are from UK, some people from Spain. So it's very interesting visualization. I think that's kind of one reason probably then PCA was very appreciated in the genetic, even with just keep the two PCs. But I think here, of course, when people realize, oh, if I have some cell types that are close to each other in the PC space, I may want to ask for more for visualization, but that's a topic we will talk about next. For now, we would say that in using PCA for single cell data analysis, most of the cases, we don't just keep two PCs. We keep more than two PCs, maybe 20 PCs or 50 PCs. So there is a range but that's about the only map. So in this case, we are just plotting the heat map for PC1. Every row is one gene. And what we are doing here is that every row is one gene. Every column is one cell. And the cells, basically the 500 cells with the largest absolute values of their scores in PC1 are plotted here and sorted by their score from the negative to the positive. And balance being true means that you have equal number of cells with negative PC1 scores versus the ones with positive PC1 scores. So like you have 250 cells on this side, 250 cells on this side. And what do we mean by PC score? Coming back here, once you have the PC1 and every cell has a projection, a position in PC1, that's the score. So for example, for the cells right here in this stretch, they have PC1 equals to 0. PC1 score equal to 0. And for these ones, they have very positive, large PC1 score. These ones have very small, right, very large negative, right? Very negative PC1 score. So that's the score. So there are two things. Just to come back, loadings and scores and PC scores. Don't confuse the two. For loading, every PC has how many loadings? The number of loadings is equal to the number of original features because every feature will have a loading to contribute to this PC. And how many PC1 scores do we have? That's equal to the number of cells because every cell will have a projection on PC1. So that's why loading is, let's say if we have n cells, p genes, then each PC has a p-dimensional loading vector. And each PC has an n-dimensional score vector. So that's the thing we should be clear about. And that will help understand the technical part. Okay, so here we are picking just 500 cells to be shown here instead of all cells. They are the columns. And the rows are the top genes in terms of loadings, absolute value of loadings, they contribute to PCA. So what we can get out of it is, again, the contribution of this MALAT1 gene and the CST3 gene is on the opposite ends, right? So one has the positive contribution, one has the negative contribution. But here, one information that's missing here, I want to let you know is that we don't have a color scale here. So it's unclear whether the yellow or the purple means larger values. That's something Syrah doesn't show you. So, but if you think PC1 is positively correlated with the CST3, and this is PC1 score, then I would guess the purple means higher value, yellow means lower value. So that's why this gene CST1 has the positive correlation with PC1. And this gene MALAT has negative correlation with PC1. And you can clearly see that the positive genes and negative genes have very distinct expression values in the cells. Right, I forgot to say here, the values are expression levels. So basically, this MALIT1 has one normalized scaled expression value in every cell. That's what I'm saying here. Okay, so I think you would intuitively argue that this PC1 is informative because we can have a very good separation of cells, right? Some cells have higher values in these genes, but lower values in these genes, and other cells are the opposite. Okay, but now you can, using this function, you can plot not just one PC, but the top 15 PCs. Still, you keep 500 cells in each heat map. What you get out of it is something very visual, because you can see that for the top 1, top 2, top 3, top 4, top 5 PCs, you see pretty clear separations of the cells. But once you go down to maybe top 14, top 15, the separation is not as clear. So this is from Surratt's explanation. The thin heat map allows for easy exploration of the primary sources of heterogeneity in the data in a data set, and can be useful when trying to decide which PCs to include for first downstream analysis. Both cells and features are genes, are ordered according to their PCA scores. But I will be more accurate. I will say cells are ordered by PC scores, features are ordered by PC loadings, right? Because you don't have a score for feature. Setting cells to a number plus the extreme cells on both ends of the spectrum, the extreme cells, the cells with extremely negative PC1 score, the cells with extremely positive PC1 score, which dramatically speeds plotting for large data sets. So this is saying that we are not plotting all cells, right? We're only selecting 500 cells to plot. Otherwise, you will have a huge heat map. Yeah. So I think this is the best figure to explain it. So PC1 is a linear combination of feature 1 and feature 2. So these two numbers are the loadings, the loading values for PC1. So this is how PC1 is constructed. And the loadings are the contributions of original features to one PC. So we will have a loading vector for each PC, and each loading vector's length is the same as the number of original features. Yeah, so loadings is only about how each PC is constructed. It's combined linearly from original features. It doesn't have nothing to do with a particular cell. Yeah, but for the scores, we are talking about individual cells. Once we have the new features as our PCs, then every cell would have a value in each PC, and that's what we call the score. So in PC1, if we have n cells, we will have n PC1 scores. Does that make sense? Okay. Okay, yeah, so just as a recap, the values here are normalized expression values of genes in cells. We are not plotting PC scores nor PC loading in this heat map at all. Okay, but the cells are ordered by their scores in PC1, and the genes are selected by their loadings to PC1 in PC1. So loadings and scores are used for selecting cells, selecting genes, ordering cells, ordering genes. That's it. And if we go back to this, we are plotting scores. For every cell, we have a PC1 score and a PC2 score. That gives the cell a coordinate. That's why we have a position for every cell. And if we go back to here, we are plotting loadings. We are plotting how much contribution each original feature contributes to PC1. Yeah. All right. Okay, so all right, now let's talk about something more technical, what PCA really does. And PCA can be interpreted in two ways. You want to find the PC, so especially PC1, first PC, you want to maximize the variance it can explain in the data. After you project your p-dimensional data to this one-dimensional PC1, you hope to maximize the variance in PC1. That's one way to interpret it. Another way to interpret it is that, let's say we have much fewer PCs than the original dimensions. Maybe you only have 20 PCs, but we have 2,000 original features. We hope that using these 20 PCs, we can reconstruct the original 2,000 dimensional matrix, and the reconstruction error is very small. By reconstruction error, we usually mean the mean squared error. We take the difference between the original, say, n by 2,000 matrix, n is number of cells, p is the number of genes, n by 2,000 matrix. We use the n by 20 PC matrix to reconstruct it. And then the reconstruction matrix minus the original matrix, we take element difference, and then we take the square of it and sum of it. If we want to minimize that reconstruction error, we can show that the projection should be PCA. So PCA is the linear dimension reduction that can best preserve can achieve the best reconstruction error. But this perspective mathematically is equivalent to maximizing the variance, which I think is something very interesting. Yeah. Okay, so we will focus on the maximizing variance perspective. So if you are interested in learning more about this linear algebra behind PCA, so I think most of the contents here can be found from the book called Introduction to Statistical Learning. It is a book we use for our undergraduate students here, and the PDF is freely online. So the authors have made the book PDF version free, so you should check it out. It's a very good intro book. And the advanced version of the book was actually published earlier. It's called Elements of Statistical Learning. So that one is very heavy, and yeah, and many PhD students might read that book. But the Introduction to Statistical Learning is for undergraduate students majoring in statistics. And also, if you want to learn more about linear algebra, because I think the interesting fact I found is that the differences in the way of teaching linear algebra is much bigger than calculus. For calculus, maybe the way we teach is more standard, but linear algebra, maybe the instructor makes a more difference in terms of whether you use geometric interpretation or you're just teaching the formula, right? So for linear algebra, I strongly recommend the course called by a professor who has a retired, Gilbert Strain. He has taught this MIT linear algebra course for many years. And the version, I think, that's most popular online is from the year 2010. So I strongly encourage you to watch it if you're interested in learning more about the topics. We'll talk like singular value decomposition, eigendecomposition, what is actually doing. Okay, so PCA is an application of eigendecomposition and SVD. So we have this matrix. Now we're going back to the more standard format, not the serial format. Pay attention. We have the N observations as rows, P features as columns. This is like the more standard format in statistics. We call rows observations, columns features, and here the observations are cells, genes are features. So we can write this matrix in two ways. We can lay out the rows. So every row is one cell, right? And we put this transpose here because the row is a row vector, and default, every vector is a column vector. So that's why we put transpose to mean it's a row vector. We have n rows for n cells, and each row is a p-dimensional vector, right? P features. Or we can write the matrix as p columns. Then every column is one feature containing n cells. The goal is to project this matrix to a new coordinate system to retain most of the variance in the n data points. So suppose that we look at the first PC. We want it to be a linear combination of the P features. So every feature has a loading, right? Because we look at PC1, then we call the loadings phi11, phi21, phi-P1. So the first sub-index is for the original feature. Second sub-index is for PC. So we have P loadings for PC1. Then you can generalize, if we look at PC2, this PC2 is called Z2, loadings become Z12, 512, 522 to 5P2, right? They are the loadings for PC2. But as a linear combination of the P vectors, each vector is n-dimensional, we get one n-dimensional vector as PC1. And this is what we call PC score. The PC score vector is here. And the loadings are the coefficients 511, 521 to 5P1. And they are scalars. They are one-dimensional. So if we look at the score for one cell I in Z1, then we call that ZI1. So it's basically a linear combination of the cell I's values in the original P features. So that's the ith row in the matrix, xi1, xi2 to xip, right? So basically, this is just a scalar form for cell I. This is the matrix form for all cells. That's the key. That's just the difference. And ZI1 is the ith element in Z1 vector. Okay, so one more explanation. While loadings are coefficients of original columns features, scores are transform rows observations. So you have one score, one PC1 score per observation per cell, and you have one loading per original feature. Okay, so again, I'm pasting this again. This is the first PC for the n observations. This is the PC1 score vector. It's a linear combination of the original P vectors. But we want to make the case simple for us. So what we did is that we basically centered our data to make sure that each column, so say this is the ith column. So that's right, this is a jth column. We have, so for the jth column here, we have the n observations sum up to 0. So basically we have xij plus x2j plus xnj, the n observations in column j sum up to 0. Just this is like centering. And we have already done that in the scale data step, right? So every column here now has mean 0 and standard deviation 1. We have already done that in the scale data step. And we also, for the purpose of identifiability, we want to find unique values for the P loadings for PC1. We require the loadings to have a square sum 1. That's what I said. It's a unit vector. So basically 511, 521, 5P1 as a P-dimensional vector, it has unit length. And that's reflected by this. Euclidean, I should say, the distance between this P-dimensional loading, if it's a point in the P-dimensional space, its distance to the origin is 1. Yeah, that's what we mean by unit vector. Okay, so for each observation I, the score in PC1 is a linear combination of its values in the P dimensions. So question for PCA to solve the problem is how do we find the P loadings 511, 525, P1 for the first PC? So our goal is to find them such that the scores Z11 to ZN1 have the largest variance. And as you know that every column has mean 0. So the mean of xi1, xi2 to xip, they're all 0. Then the mean of zi1 is also 0, right? When we know the mean is 0, then the variance formula is just zi1 minus the mean minus 0 squared. So then it's just zi1 squared. This is the variance of the n cells, n observations in PC1. And because of this formula, we can write zi1 as linear combination of these xij, right? J from 1 to p. So it's just some representation of the same thing. So from here down to here, that's what we have. And then we have this constraint. The constraint is that the loadings should have their square sum up to 1. Okay, so that's the optimization problem. Basically, we're trying to find 511 to 5P1 by maximizing this subject to this constraint. Okay, then we use matrix notation. So basically, It's the same thing, but we just write it in the matrix form. So we denote phi1 to be this p-dimensional loading vector, phi1, 1 to phi p1 for PC1. Then this term is essentially, if we drop this 1 over n, okay, because 1 over n doesn't depend on the features. It's just the sample size, number of cells we have. So it has nothing to do with our search for phi1, 1 to phi p1. So we just look at the summation zi1, I from 1, zi1 square, I from 1 to n. You can show that it's basically z transpose z. Z is what? All right, so I should make a correction. Yeah, I should make a correction because I now change this to z1. So this should be z1. Okay, this should be z1 and this should be z1. But basically, if you use the notation here, so z times x phi 1, this is already our PC1. Then basically, the sum of squares is just the inner product of the vector to itself. So because z transpose here is 1 by n, a row vector, z is n by 1. So basically, multiplying them together, you get a scalar, and that's the sum. And then because of this formula, you put it in, right? Z is x times phi 1. Then z transpose becomes phi 1 transpose x transpose, then times z x transpose phi 1. That's what we have. Then you're trying to maximize this term. Is still, the product is still a scalar, but it's a matrix multiplication subject to the same constraint. So I just want to say that this line and this line is the same thing. The only difference is that this is like the scalar form, this is the matrix form. But if you are okay with the matrix form, the calculation is much simpler. Yeah, you can simplify a lot of things. And so for this objective, how do you solve it? And this becomes an optimization problem. In the optimization problem, a very classical technique is to incorporate this constraint into our optimization. When this happens, right, then if you take phi 1 transpose phi 1, so by the way, I should also add that phi 1 transpose phi 1 is equal to the sum. Basically, it's equal to the sum of its square, element y squared, because it's in the same vector multiplying itself again. So if you're not familiar with this result, just try to do the vector, write it out. Phi 1 as, you write out phi 1 as phi 1, 1 to phi p1. You do this vector transpose times vector, and you will see it's exactly equal to this term. Okay, then with this constraint, you put it inside your objective because you know it should be equal to 0, right? When it is satisfied. So you put it in, and this is called the Lagrange multiplier into this, but you introduce an additional parameter called lambda, and to maximize it jointly, because you use minus lambda, right? So you see in this new optimization problem, since it's minus lambda, you want to encourage this term to go to 0. And this is exactly the constraint we want to achieve. And then for this optimization, it's something we learned from calculus. If you want to optimize something, you take the gradient or derivative, set it to 0. And here, what we want to optimize is the phi 1. That's the argument, the value we want to search for. Then we take the gradient against phi 1 and set it to 0. So this is like a vector calculus because we're taking a gradient against a vector, not a scalar. So we know the result is 2 times this thing times phi 1, because you have 2 phi 1, one on the left side, one on the right-hand side. Once you take the gradient, you get a 2, and you only keep the right one. And for this one, you get a 2 lambda, the right one, and this constant becomes 0 once you take the gradient. When you set this to 0, you move the term to the right-hand side, rearrange the terms, cancel 2. This is what you have. And this is the very nice result because this is exactly the definition of an eigenvalue. If you have ever taken a linear algebra course, right? It's a matrix times a vector equals a constant times a vector. Then this constant is called the eigenvalue. And correspondingly, the vector phi 1 is the eigenvector. Okay, so basically we show why we can change the PCA problem to an eigenvalue problem. And because we want to maximize, right? Don't forget, we want to maximize this But this is to be interpreted, this is interpreted as maximizing the variance. For this purpose, we want this term to be as large as possible. And if we multiply, go back here, right? We have this term equals lambda times phi1. But now we want to left multiply phi1 transpose here because that's the term we want to maximize. When you left multiply phi1 transpose here, you also left multiply phi1 transpose here. And lambda as a constant can get out of it. And phi1 transpose phi1 is 1. So essentially, you have this thing to be maximized equal to lambda. Since you want to maximize it, and it's equal to an eigenvalue of this, then naturally, we want to look for the largest eigenvalue among all possible eigenvalues. So another result is that the number of non-zero eigenvalues is equal to the rank of your X matrix, the rank of your X matrix, because if you have, say, P columns, P features, and when n is larger than P, then we say this matrix has full column rank, right? So in this case, we could, I shouldn't say that, but in general, if you don't have perfect correlated columns, I should take it back. Then you have full column rank, then you should have P non-zero eigenvalue, P positive eigenvalues. But if the matrix has some other issues, maybe some columns are perfectly correlated, or if you have fewer rows than columns, more variables, fewer observations, then you will not have full column rank, right? So basically, there's an interpretation for number of non-zero eigenvalue, that non-zero positive eigenvalues. But here we are looking for the largest eigenvalue. That's the lambda. And then the corresponding eigenvector is the phi1. So basically, in short, the loading vector of PC1 is the eigenvector that corresponds to the largest eigenvalue of this X transpose X. And this X transpose X has an interpretation. It's P by P. P is number of features. So this is called sample covariance matrix of the P features. And because we already have centered each column to have mean zero, and here we have standardized each column to have standard deviation 1. I can even call this sample correlation matrix, because once you have standard deviation equal to 1, variance becomes correlation. That's another thing. Well, I'm saying here, but I'm not writing down the detail. Yeah. So there are a lot of steps to fill in, but I hope to give you the general idea that why PCA is basically doing eigen decomposition on this X transpose X. How about PC2? If we want to find the PC2, and we call that Z2, it's again the same P features, but now linearly combined using the second loading vector, right? We are looking for the second loading vector. Again, we want to maximize the variance in the second PC, but we have one more constraint. So besides requiring that the second loading vector has unit length, we also need it to be orthogonal to the first loading vector. So phi2 transpose times phi1 is equal to 0. We need to add this additional constraint. And then we can show that we would just need to have phi2 as the eigenvector corresponding to the second largest eigenvalue of this matrix. So basically, the eigenvectors for different eigenvalues are orthogonal. That's a nice property for eigen decomposition. Finally, how many number of PCs can we have if we don't stop? We just go to next PC, next PC. It's the minimum between of these two numbers, n-1 and p. So if your n is very large, then it's just p. You can have p original features, p PCs, right? But if you have very small number of observations, and in statistics, we call that sample size n, then you may not be able to go that far. But in our single cell data, I don't think that's the problem, especially that we only keep 2,000 genes in the previous step, then you will most likely have more than 2,000 cells, right? Otherwise, you cannot do that. Okay, so to quickly summarize, the essence of PCA is eigen decomposition of X transpose X, the sample correlation matrix of the P genes. You kept the 2,000 genes, P to 2,000 genes you kept from the highly variable gene selection, and you're using the normalized data that's centered and scaled. And this eigen decomposition can be done via singular value decomposition of this matrix. So that's the thing I will not talk about. But basically, it's a very standard linear algebra technique. Something good to know is that for singular value decomposition, the matrix M by P doesn't have to be square or symmetric because it's M by P, right? In contrast, this X transpose X is P by P, and it's symmetric and it's square. And so eigen decomposition can only be applied to such a matrix. But singular value decomposition is very general. It can be applied to any matrix. So you would have left singular vectors, right singular vectors. What we need is the right singular vectors, which is equivalent to the eigenvectors of X transpose X. But the left singular vectors of X is equivalent to the eigenvectors of X times X transpose. So they have this correspondence. And also between singular values and eigenvalues, they have a square relationship. Like the eigenvalue is the square of singular value. So these are just the linear algebra facts. But basically, if we don't have a large computation issue, we just use singular value decomposition. And this will give us exact PCA. And how do we evaluate the PCA results? The most important criterion is the proportion of variance explained. So for each PC, we can calculate how much proportion of the original data's variance explained by it. So we have this ratio. The denominator is just the total variance of original data. What we did is that we take each feature j, we take xij squared, because feature j, basically sum of in that feature j, and then we take the sum across all features. That's the total variance. And the top, the numerator, is the variance of the scores in the mth PC. So m could be 1, could be 2, could be up to the total number of PCs. So then we have this linear combination as the scores in our mth PC, right? This is just the PC scores of our observation I in the mth PC. That's it. And then we take the square for each observation, sum over observations I from 1 to n. That's the proportion of variance in the mth PC. That's it. And we have this fact that if you go through all the PCs to the maximum number of PCs, you add up the proportion of variance explained. So these share the same denominator. They only differ in the numerator because the numerator depends on the mth PC. Denominator is the same. They add up to 1. And another property is that PVE1 is the largest. It's larger or equal than PVE2 and so on. The reason is that when we look for the PCs, we intentionally find PC1 to maximize the variance. That's just the simple reason. Okay, so therefore, later we'll show you that if you plot the PVE for each PC along with the PC from 1 to 2, you will see a decline curve. That will show you how fast does the PVE decline as you include more and more PCs, right? Where should you stop? Basically, intuition is that if up to some PC, say PCM, the PVE of PCM is very small already, maybe we don't need it. Okay, and this is a very nice paper my student Zhu Tian sent to me. It's a review. Basically, it's a benchmarking PCA for single cell RNA-seq data, which I will post. Because single cell RNA-seq data has some unique challenges. For example, you may have many cells. Like from this paper, I can quickly show you. The paper used 4 data sets. The number of genes is actually roughly on the same order, right? But the number of cells can go from 700 to 130K. So you see that given the cell number increases that much, the file size, data size can be much larger, from 24 MB megabytes to 58 gigabytes. So when you have 58 gigabytes, it's most likely that you cannot do the PC on your laptop. because our laptop's RAM may be less than that, right? Even if 64 gigabytes RAM, you don't want to use it just for the PCA calculation. So that's why for such data, we need some efficient PCA algorithms for memory and for computational time. So that's why in this paper, you see how many algorithms are there. So this paper compares these many algorithms and say that different algorithms have different theoretical properties, implementation in R or Python or Julia, and they evaluated what downstream performances of PCA and availability and so on. Yeah, so basically it's a very comprehensive benchmark of PCA algorithms. But let's go back to this. So maybe we can just read what's in the paper. PCA is widely used for data visualization, data QC, quality control, feature selection, denoising, imputation, confirmation removal batch effects, confirmation estimation of cell cycle effect, rare cell type detection, cell type and cell state similarity search, pseudo-time inference, and spatial reconstruction, right? So basically listed all the downstream things you could do after PCA. Additionally, PC scores are also used as the input of other nonlinear dimension reduction, like t-SNE, UMAP, and clustering methods in order to preserve the global structure, avoid the curse of dimensionality, because you only work with 20 to 50 PCs instead of 2,000 genes, and save memory space. A wide variety of single-cell RNA-seq data analysis tools actually include PCA as an internal function or utilize PC scores as input for downstream analysis. We review the existing PCA algorithms and implementations and classify the algorithms into 6 categories, namely similarity transformation-based, SIMT, downsampling-based, DS. By downsampling, basically saying we downsample the cells observation, so we work with a smaller m. That's downsampling. Singular value decomposition update-based, this Krylov subspace-based, gradient descent-based, and random projection-based. So these are all different implementations of the optimization. So I would say if you are someone who is working on statistics machine learning these days, knowing optimization is super important. Otherwise, the algorithm can take super long to run or even unable to run on big data. So that's why I think optimization traditionally offered as a double class has become more and more popular among statistics machine learning students. Yeah, that's just the background. We have listed 21 PCA implementations comprising 10 algorithms, because every algorithm may have more than one implementation that are freely available and easy to download, install, and use for analysis. The correspondence of the review PCA implementations and single-cell RNA-seq studies is summarized in Table 1. Just for information, as ScanPy is in Python, SURAB is in R, they actually use slightly different PC implementation. Yeah, that's something good to know, because that can be one small step that contribute to their differences. But there could be differences in other steps. So that's why it has been recently discussed that their cumulative differences in the results can be large, depending on the data set for sure. But this is from this paper back to 2020 genome biology paper. I'll post that. So this is how to say single-cell RNA-seq methods, and their PCA algorithm. So which one do they use? So you will see in most cases, for most studies, they use the standard PCA. So in R, the standard PCA, the exact PCA is done by this command prcomp, principal component, or SVD. So they are equivalent in R. That's the exact PCA. In Python, sklearn, you have PCA. This is the standard slowest method. But if you go down to ScanPy, right, it uses this one called IRLBA. Even though it's also in the Python PCA function, but you need to specify it. So you can specify it to be the standard way, this one, or the IRLBA. You have the option, and IRLBA is faster. Okay, so just for your information, ScanPy is not using the exact PCA. Okay. Yeah, and also like methods like Harmony for integration also uses IRLBA. Yeah, so just for information that there are 10 algorithms here. And specifically, I don't know if it's too small, but I think it's worth seeing that you would need to look at the time complexity, memory complexity. These two theoretical properties are super important. So for information, the standard PCA here, or the PR count, right? These are the standard PCA. The time complexity is n times the minimum of n and m. What is n and m? N here in the author's notation, n is number of genes, m is number of cells. So it's most like you need a square of one term, either the square of gene or square of cells times the other one, right? So if it's OM square, then it's the number of cell square times the number of gene if the cell number is smaller. Otherwise, it could be n square m if the gene number is smaller. So basically you have a square term here. But if you go to this IRLBA, it's just K times M. M is the number of cells, and K is what? K is the number of PCs you want. So you pre-specify how many PCs you want, and we just go there. We just truncate our results there. We don't go through the full PCA. That's why it's faster. Yeah, but it's an approximation. It's not exact. Okay, and also, do you have those, how to say that, parallel computation or online learning methods? So this is OOC PCA. Let's look at here. So here, or this incremental PCA, these two, they have this additional advantage besides the time complexity. You have the additional advantage of this B times M. What is B? B is the block size, number of sample genes. So when you do this incremental PCA, you're not running on your full matrix. You divide your original genes features into blocks. And then this block size B, maybe say 20, 50, that becomes the key factor in determining the memory usage and time complexity. And that's very advantageous. But still, you lose some accuracy. But when your data is so large, that's better than, it is found by this paper, it's better than the downsampling cells, which is something I think good to know. Okay, so we'll quickly wrap up and show this, which is also from the paper, and it's very interesting. So you see that using different PCA implementation, this is the exact one. This is the truncation one, fast truncation one. But the truncation one, the IRLBA, so the memory usage is still n times m, number of genes times number of cells. So you may have the memory issue here, even though it's fast. So that's why for the two larger data sets, these two about brain, they have more cells than the first two data sets, PBMC pancreas. You would have memory issue with the standard exact PCA and the IRLBA PCA. They cannot give you result. But downsampling, incremental, in both like incremental here, this and this, they are this online learning version, which I will introduce later. Yeah, so they can give you a result. But the difference here is that this is by Julia implementation, which is another language different from R and Python. Those two have very good computational efficiency. They can give you results. But be careful about the differences of the results. For example, we consider the exact PCA to give, and this is probably, I think, TSNE or UMAP thing, right? You use that for TSNE or UMAP. The exact PCA is considered to be the gold standard here, the baseline. You see that the purple and the blue clusters became merged or connected if you use downsampling. So you change the result here. And here also, you have these two become closer after you do downsampling. So basically, downsampling is considered to be the bad practice by this paper. If you have a large data set for you to run this, don't use downsampling, but consider online algorithms or this IRLBA. So we will stop here from this result. This is like just the guidance for users. I think it's very informative. It really, your choice of the algorithm depends on this product, number of genes times number of cells, called GC. So when the product is less than 10 to the 8th, when your data is not so small, you could use exact PCA. Yeah, you could just use exact PCA, no problem. But of course, it depends on whether your data is dense or sparse. So, we mentioned the sparse matrix coding, right? In Sura has that. So you can save your memory, save your computation. So in general, sparse matrices are better for computation. And for single cell data, since we have many, many zeros, we might be able to do sparse matrix. However, be aware that we have done the normalization and we have done the scaling. So probably data is no longer sparse. And this is something you could check. And I think it's no longer sparse. Anyway, so this GC is the key thing to think about. If it's small, you less than 10 to the 8th, do exact PCA. Otherwise, use the IRLBA, right? That's the truncated version. If it's greater than 10 to the 10th, you cannot do much, then do the out of core approaches, like the online learning approaches. So we will come back to that next time. All right, so I'll see you on next Monday. Hello. Okay, let me try to stop the recording.